and welcome to the Absolute Recap Biology Edition, where we aim to maximize your understanding and minimize your need for memorization. Each episode will recap content, skills, and test-taking tips to help you succeed in May. I'm your host, Melanie Kingett, and your recap starts now. Hi, and welcome to the Absolute Recap Biology Edition. Today's episode will recap active transport. Let's zoom out. We're in Unit 2, Cell Structure and Function, topics 2.6 and 2.9. Our big idea is energetics. There's no room in the Uber, but your friend begs the driver, "Please, can't we just fit one more person? Look, we will all just scooch over," as he hops into the back seat, stepping on toes and struggling to close the door.
Dude.
It's uncomfortable for everyone. Just like you, molecules need their personal space. Well, at least as it pertains to molecular interactions. And they will not crowd themselves without some effort.
How's that for roominess?
And since in biology, these molecules are typically contained in cells, that effort will often come from good old adenosine triphosphate. Let's zoom in. There are two types of cellular transport, passive and active. These categories are divided by the use of energy and direction of molecular movement. As we've discussed previously, passive transport is the net movement of molecules from an area where they are more highly concentrated to an area where they are less concentrated, without the input of metabolic energy, or ATP. For a full recap, check out episode 53. It's really important that cell membranes establish and maintain their internal environments through selective permeability. After all, homeostasis is the maintenance of constant internal conditions despite external changes, and environments are always changing. One of the ways cells accomplish this is through the strategic movement of molecules by active transport. So, how does the cell move molecules against the concentration gradient, especially those that are large or charged? The movement of molecules from regions of low concentration to high concentration is known as active transport and requires the spending of cellular energy, or ATP. This movement is often referred to as up or against the concentration gradient, but be careful with the word up.
Now listen carefully.
There is no up, down, left, or right in the 3D world of cells. Instead, we will want to use the phrases like into the cell or out of the cell to reference the movement of solutes. There are three main types of active transport: membrane pumps, endocytosis, and exocytosis. Let's start with membrane pumps, which are transmembrane carrier proteins, just like with those used for facilitated diffusion. But since this is active transport, the molecules move against their concentration gradient and use ATP. Transport proteins are solute-specific due to their unique chemical folding and R-group interactions. When ATP is hydrolyzed, a phosphate group is removed and often attached to the pump itself. Phosphorylation causes a conformational change in the protein's shape, adjusting the microenvironment within the protein channel and shuttling the solutes across the membrane.
If you were to find yourself faced with the opportunity to hunt for buried treasure, and you had the option of either A, going on that treasure hunt with an experienced adventurer and access to the treasure map, or 2, winging it and just figure it out as you go. Now, winging it is admittedly very exciting, but the phrase, "I'll figure it out as I go," is usually the last thing I say before getting very lost or spending hours digging through the trash for the furniture assembly instructions I said I didn't need. An experienced adventurer and access to the treasure map is probably the smartest option. Now swap out the word treasure map with study guide, adventurer with teacher, and buried treasure with educational aspirations, and I don't know where I was going with that. But what I do know is that we have study guides, virtual classrooms, and tutoring sessions available now at www.TheApsoluteRecap.com. Now back to the recap.
One of the most common examples of active membrane transport is the sodium-potassium pump, which performs a crucial job in the nervous system to maintain membrane potential, or charge. The sodium-potassium pump moves 3 sodium ions out of the cell for every 2 potassium ions moved into the cell, in somewhat of a seesaw motion, where the protein is open to one side of the membrane at a time. How can there be a difference in membrane charge if these are both positive ions? It comes down to ratios: 3 out for every 2 in. So the extracellular environment has a net positive charge. Another example is the proton pump. And spoiler alert, it moves protons. Proton pumps are used during the electron transport chain in chloroplasts and mitochondria to establish an electrochemical gradient of hydrogen ions. Both the sodium-potassium pump and the proton pump work to accumulate ions on one side of a membrane, which the cell may later use during cotransport or with ATP synthase. Molecules rarely stay concentrated for long. The two other types of active transport involve vesicle formation for endo and exocytosis. Since all membranes are primarily composed of phospholipids, the formation and fusion of vesicles is a fairly seamless process.
How did that get in there?
Endocytosis is the process of bringing substances into the cell in larger quantities and has 3 types: phagocytosis, commonly referred to as cell eating, penocytosis, or cell drinking, and receptor-mediated endocytosis, where specific ligands bind to cell surface receptors, causing vesicle formation. Receptor-mediated endocytosis is the pickiest of the 3. Remember the endosymbiosis theory? Well, the foundation of that theory is phagocytosis, the engulfing of a small prokaryote, resulting in the double membrane energy transducers like chloroplasts and mitochondria. Exocytosis involves the fusion of a vesicle with the plasma membrane for molecules to exit the cell. These might be intentional products that were shipped out by the Golgi or waste products that the cell is removing. Time for unit connections. Active transport is involved in Unit 2 with the endosymbiosis theory, Unit 3 with photosynthesis and cellular respiration, and Unit 4 with cell communication and signal transduction. All right, what about the exam? You may be asked to predict molecule movement based upon experimental design, draw conclusions about concentration gradients, assign experimental questions to membrane protein models, describe the pathway of vesicle movement, or to describe the movement of molecules to a new scenario, like absorption in the human intestine. To recap, active transport moves molecules against their concentration gradient from low to high with the use of ATP. The 3 types of active transport are membrane pumps, endocytosis, and exocytosis. Before we conclude this episode, I want to give a shout out to some of our most active Instagram followers: Tiffany Jones of AP Bio Penguins, Jose Sanchez, Jess Snowden, and Joanna McLean. You are the absolute best. Thank you so much for following along, engaging in our posts, and spreading the word about our podcast, products, and services. Remember, we currently produce recaps for 5 courses: chemistry, U.S. government, music theory, physics, and of course, the OG, biology. Coming up next on the Absolute Recap Biology Edition, glucose combusting. Today's question of the day is about transport. How is a cotransporter classified that moves molecules in opposite directions? For the answer to the question of the day, please follow us on Instagram at TheApsoluteRecap. That's the APsolute Recap. Check out our website, TheApsoluteRecap.com, for episode schedules, study guides, virtual tutoring, and to sign up for our virtual classroom. The Absolute Recap is produced by Brad Kingett with music by Zach Caruso. Today's episode was written by me, Melanie Kingett. Thanks for subscribing, and don't forget to rate and review wherever you get podcasts. Time's up, pencils down. Thank you for listening to the Absolute Recap Biology Edition. AP is a registered trademark of the College Board. Copyright 2020, Absolute Recap LLC, all rights reserved.